I use CRISPR primarily to understand how genes activity are regulated in our genome. So it's kind of a repurposing of the original technology in a way that we can now use it to get to the nitty-gritty details of the activity of the genome. There have been a lot of excitement in the field in the last, I think it has been now almost 10 years in the field about using CRISPR, which is a tool that by which basically you can cut a specific part of your genome, and that basically allows you to delete genes that may not be useful or may cause diseases. What I did was basically we used a version of it that basically doesn't cut at all DNA, but we used it to basically understand the regulatory part of the genome, meaning we use that to understand the switches that controls the activity of the genome. So this is a version of the CRISPR that doesn't cut. It's called deactivated CRISPR or Cas9. And we basically use that to compete with the molecules that binds to our genes and controls the activity. And by removing them, we can now basically explore this unexplored area of how interactions between proteins and DNA control activity of the genes. And that's pretty much, relatively a new area to go to the not genes, but what is known as regulatory elements of the gene and understand how they switch on or off the genes that we are studying or have been studying for a long time. If we know the switches that turn the genes on and off, we have a better shot at trying to control those genes that are involved in diseases. If we can, if there is a gene that is specifically involved in a disease, instead of going and trying to delete the gene, if we can control their activity, we have a lot more options in front of us to basically approach diseases. What I would consider, a career that was well done and well performed, I think would be one in which I would be able to answer this question of how cells think, how they process the signals that they get from the environment. In particular, in the context of stem cells, stem cells, they have many choices. They can become a neuron, they can become a fibroblast, they can become blood cells. So they have all these options. And how do they compute the signals from the environment in order to make the right decision? I think that's sort of the inner working within individual cells of how they process the signals from the environment to basically coordinate these important decisions, such as whether I should divide right now or no, I should differentiate or no. Those are the questions that sometimes keep me awake during the night.